Hi everybody, welcome to Applied Stem Cell webinar. This webinar is on TARGATT, which is rapid and efficient transgene integration technology, particularly on stable cell line development and generation of large mammalian cell-based screening libraries. By far, the libraries on the market are mostly generated from bacteria, yeast, and mammalian. For the libraries generated from the bacteria, it's cost-effective and it's fast to generate large-size libraries. But the drawback is there's no post-translational modifications. For the library generated from yeast, the environment is eukaryotic, but it only contains some parts of the post-translational modifications, not all of them. As for the library generated from the mammalians, and it's close to full human environment, and also it contains all the post-translational modifications. And the mammalian host can definitely help to capture relevant biology elements, for example, promoters, enhancers, and splicing components, which is very unique to human. But the drawback for mammalian library is the process is slow and is very laborious. So as for now, the size varies a lot among different types of libraries. As shown here, the library size for both bacteria and yeast can reach up to 10 to the 8th to 10 to the 9th. However, as for the mammalian library, the size is limited to only 10 to the 4th to 10 to the 6th. And this also involves a lot of labor by making plates from 20 to up to 200 plates. The major reason for small-size mammalian library is due to the low knockin efficiency of inserted genes. By using our TARGATT technology, our goal is to be able to hit within an order of magnitude typical for bacteria or yeast libraries. So finally, the library size we expect will be around 10 to the 8th to 10 to the 9th. For our TARGATT technology, we have the exclusive rights from Stanford. This shows how TARGATT works. There's an attP site pre-inserted in the genomic DNA of the cells, and there's an attB site in the donor DNA together with the GOI. The integrase will drive the recombination happening between the attP site and also the attB site. Then the GOI, the gene of interest, can be inserted into the locus. So the integration system here showing is site-specific, irreversible, and it works in non-dividing cells. The knockin size for genes can be large, up to 20 kb. Some key features listed here for our TARGATT screening system. First is copy number controlled, which is 1 copy per cell. And the second is low biosafety level, which eliminates the need for virus-based libraries. And the third is the integrase efficiency is high, can up to 10 to 30%, because the efficiency will impact the library size a lot. And due to the defined locus, there's a consistent expression across construct. And finally, the protein expression level is medium to high, which meets the R&D level of protein production. Here shows the system that we have generated for site-specific transgene integration. We have validated in several cell lines, HEK cells, CHO cells, and this shows how TARGATT works here. So the master cell line contains 2 attP with landing pad contains other stuff showing here, puromycin, thymidine kinase, and also 531 for different purposes. And in the donor DNA, there's 2 attP sites and 1 GOI, and also bacterial backbone containing the TK as well. So after recombination happens, either the GOI will got inserted or the TK with bacterial backbone got inserted. Drug selection was performed after to eliminate the TK positive cells, which including the parental cell line and also the bacterial backbone cells. So the final population in the cells will be only contains the gene of interest. That is our goal for this system. To make the master cell line with docking site ready at target system, so we tried to our benchmark locus at H11 first. So here shows how the master cell line development. Firstly, CRISPR-Cas9 technology was used to generate the master cell line with landing pad inserted at H11. The landing pad, as I described earlier, contains two attP sites, puromycin, TK, and 5C31, all driven by PGK promoter. For checking the right site-specific insertion, it was done by junction PCR of 5-arm and 3-arm. And also ddPCR was used here for assessing the single copy numbers for the landing pad. So after donor DNA knock-in and undergo the drug selection, we can see here from the CHO cell lines, the GFP positive cells enrichment was achieved with up to 90% in 2 weeks. And this is confirmed by fluorescent microscopy by comparing the random insertion and the site-specific insertion. And FACS analysis showing the integrase efficiency is up to 13% before GCV selection and 97% after GCV selection. And also, Western blot was used to confirm the GFP protein expression, expressed well and got enriched after GCV selection. To confirm the single copy insertion, the cells was knocked in by 2 donors, which contains GFP and M-cherry separately. If the system is single copy, only GFP or M-cherry will be knocked into the individual cells, and only green or red cells can be observed in the population. As shown on the left side, under microscopy, on day 2 transfection, you will see a yellow color from the cells due to the transient expression of GFP and M-cherry. However, after 10 days of transfection, with transient expression cells went away, only cells with either green or red cells were observed. After selection, the green or red cells were greatly enriched by itself, but no yellow color cells were detected. This suggests the system is one copy system, and this single copy insertion was further confirmed by FACS analysis showing on the right side. So H11 is our benchmark safe locus and is located in chromosome 11 between the 2 highly expressed genes as labeled here. Insertion at H11 resulting in little or no phenotypic change, and its activity has been confirmed in mouse, pig, and CHO cells using GFP as a fluorescent marker. And the protein expression level at H11 is medium to high, as confirmed by 2 antibodies with about 1 gram per liter after 11 to 17 days of fed batch culture. To further evaluate the docking site, several other locus were found and assessed. As a result, LC2 outperformed other locus in expression levels and expression stability. As shown on the left side, when using GFP as a marker, ASC2 site showed the highest GFP expression level and kept stable over 82 days. Comparing to the H11 site, as the data showing on the right side, ASC2 showed over twofold GFP expression level higher than H11 after 56 days post-transfection. To move further, related to more relevant proteins, at these two locus, an Fc fusion protein was cloned into the donor DNA vectors driven by the CMA promoter. And the whole process only took about 1 month to get to the final step for analyzation. So firstly, the cell was expanded at day 0 and transfected with donor DNA at day 7. After selection, seed flask at day 27 and protein expression level were analyzed at day 33. The titers from 6-day Shake Flasks by HPLC showing 2 clones with about 142 milligram per liter from ASC2 sites and 2 clones with 10 to 18 milligram per liter from H11 sites. This confirmed that the protein expression level is higher at ASC2 sites than H11 site. Then we further pick clone 42 for further subclones, and the expression level showed very consistent within the 20 subcloned cells, and the average clone's titer is about 14 milligram per liter. This suggesting the system is very stable and robust. To compare the target system over other systems on the market, the advantage for target is site-specific, irreversible, and has controlled copy number, and efficiency is high. As for recombinase, it's site-specific insertion. However, it's reversible insertion, and copy number is depends. The efficiency is very low. And as for the transposes, the integrase knockin efficiency is high. However, it's random insertion, and the copy number is very variable. So our target screening system can be used for 3 major platforms. The first application is immuno-oncology, which is used for antibody discovery and affinity maturation. The second application can be mammalian protein evolution for expression elements, protein function optimization, and AAV capsid libraries. The third application can be CRISPR library screens. By using targets, no need to worry about the MOI, and it can help to reduce the biosafety level by eliminating the virus handling, and also it can help to reduce the workflow requirement by eliminating the virus packaging. So this is a webinar focused on stable cell line development and screening libraries. If you want to get more information, please feel free to reach our website. And also, we can be reached by email or phone. Thank you very much for your attention.